Welcome to the deep dive. Today, we're tackling a really widespread issue, inflammation and edema.
Mm.
I mean, just think about how many people you know, or maybe even yourself, who deal with the aches, the swelling, like with arthritis, for instance.
Yeah, it's a big one. It definitely impacts quality of life.
Hugely.
Oh,
So that's why we're taking a close look today at some, well, frankly, fascinating science. It's pointing towards a really promising approach, photobiomodulation therapy,
Ro-
or PBM.
PBM.
Our goal today is pretty simple, really. We want to explore the powerful research that's showing how specific types of light can actually help our bodies fight back against that inflammation and, the fluid buildup.
And we're focusing specifically on the positive findings here, the compelling stuff.
Exactly, just the
Yeah.
positive, exciting research in this area. So maybe start us off, inflammation, edema, what are we actually talking about?
Okay. So inflammation, fundamentally, it's the body's initial reaction to trouble,
Yeah.
right? Injury, infection.
Mhm.
It's like sending in the first responders.
Makes sense.
But the problem comes when it hangs around too long, becomes chronic, or it's just excessive.
I have.
That's when it causes damage.
And the edema, the swelling.
Well, that often goes hand in hand. It's basically fluid leaking out. The inflammation can cause imbalances, pressures change, and fluid escapes from blood vessels into the surrounding tissues.
Okay, that uncomfortable puffiness.
Exactly that. It involves proteins and fluids leaving the vessels.
And, in research, I've seen they use models, like that carrageen-induced paw edema in mice.
Yes, that's a common one. It's a way to mimic that acute inflammation. You get the swelling, the edema, and also hyperalgesia.
Which is that increased sensitivity to pain, right?
Precisely. Increased pain response. So that model helps scientists understand the basic mechanisms.
And how that relates to things people actually suffer from, like, say, rheumatoid arthritis.
Exactly. RA is where the body's immune system mistakenly attacks the joints. You get this cascade of inflammation, cytokine release. It's quite destructive.
Or osteoarthritis, which is more wear and tear.
Right. OA is more degenerative, cartilage loss, bone spurs. But inflammation is still a key player there, too, in the synovium.
And the current treatments, drugs and so on, they help, but they often have, well, downsides, side effects, limitations.
That's a major issue, yeah.
Yeah.
Finding effective and well-tolerated solutions is really the goal, which brings us back to PBM.
Right. Photobiomodulation. So this is using light as a therapy.
Essentially, yes. We're talking about specific wavelengths of light, usually in the red and near infrared spectrum, often delivered using lasers or LEDs.
Okay.
And the key thing here, it's not primarily about heat. It's not like a heating pad. It's about triggering a photobiological response within the cells.
A biological response. So the light actually changes how cells behave.
That's the idea. It modulates their function.
Yeah.
And crucially, based on a lot of research, it seems to help reduce that harmful inflammation.
Okay.
There are quite a few studies now, both animal experiments and clinical trials, reporting these anti-inflammatory benefits, especially for conditions like arthritis.
Okay, so how does that work? I mean, how does shining a light, actually make a difference down at the cellular level? That's the really intriguing part for me.
It is fascinating. So think about the power plants inside our cells, the mitochondria.
Right. Generate energy.
Exactly. Well, within the mitochondria, there's a key molecule, an enzyme called cytochrome C oxidase. Let's call it CCO.
TCO. Okay.
Now, CCO is what's known as a chromophore. It's particularly good at absorbing light in the red and near infrared range, the very wavelengths used in PBM.
So CCO is the target for the light.
It's considered a primary photoreceptor, yes.
Yeah.
When it absorbs these photons of light, it seems to enhance mitochondrial activity. This can lead to increased ATP synthesis.
ATP being the cell's energy currency.
Precisely. More ATP means the cell has more energy, it can function more efficiently, potentially repair itself better, and just be more resilient overall.
Okay, so boosting the cell's energy helps it kind of calm down inflammation. That connects. Now, what about, I've heard about reactive oxygen species, ROS. Aren't those usually the bad guys? Oxidative stress and all that?
They can be, yes. Oxidative stress is definitely linked to inflammation and damage. But here's where PBM gets really interesting. Its effect on ROS seems to be, well, biphasic. It depends on the dose.
Biphasic,
Meaning that while very high doses of light might actually increase harmful ROS, optimal therapeutic doses seem to generate small, transient amounts of ROS that act as beneficial signaling molecules.
signaling, like telling the cell to do something good.
Exactly, like a little internal alert. These beneficial ROS can activate pathways involved in healing, reducing inflammation, and protecting the cell. So paradoxically, the right dose of PBM can actually help reduce overall oxidative stress in diseased or injured tissue.
Wow. Okay. So it's not just blasting away. It's about hitting that sweet spot, that Goldilocks zone to get the good kind of signaling.
That's a great way to put it. It's about modulation, not just brute force.
Okay. Now let's talk about the actual messengers of inflammation. Things like TNF alpha, IL-1 beta, IL-6. These cytokines are major players in arthritis, right?
Absolutely key players.
Right.
These pro-inflammatory cytokines drive a lot of the destructive processes and symptoms in conditions like RA and OA.
So how does PBM affect them?
Well, this is where a lot of the compelling evidence lies. Numerous studies in animal models of arthritis and even in vitro studies using human cells have shown that PBM can actually downregulate these pro-inflammatory cytokines.
Downregulate, meaning it turns down their production,
Right.
reduces their level.
Yes. It can decrease the expression, the amount of these key inflammatory signals. Essentially, it helps quiet down that inflammatory storm.
That's significant.
It is. And it's important to note the specific effects can vary depending on the PBM parameters used, the wavelength of light, the dose or fluence, the power density, all these factors matter.
Right. Which brings us nicely to some specific wavelengths, particularly the ones relevant to MedWave's work. 810 nanometers and wavelengths around 635 nanometers. What's the science say about those specifically?
Okay, let's take 810 nanometers first. There's research indicating that 810 nanometer laser light can lead to a reduction in the mRNA expression. Basically, the genetic instructions for pro-inflammatory cytokines. And this effect seems to be fluence dependent.
Meaning the amount of light energy delivered matters.
Correct. More light within a certain range might lead to a greater reduction. And interestingly, one study used an 810 nanometer laser in a mouse model, actually of Alzheimer's disease, and found it upregulated beneficial signals like IFN gamma and IL-10.
Anti-inflammatory signal.
Generally considered more regulatory or anti-inflammatory in that context, yes. And this correlated with cognitive benefits in the mice.
Wow.
And another example, in a rat model of TMJ inflammation, that's the jaw joint 810 millimeter laser treatment, was shown to reduce levels of pro-inflammatory cytokines in the joint tissue.
So 810 seems pretty solid for modulating inflammation in different spots.
The evidence points that way, yes.
Okay. What about the other range, around 635 millimeter, or maybe 660 millimeters is often mentioned too?
Yes, that red light range is also very well studied. Remember that Alzheimer's mouse model study?
Yeah.
Well, it also found that 635 millimeter light could boost those potentially beneficial signals, IFN gamma and IL-10.
Okay. Similar effect to the 810 in that case.
In that particular study, yes. And then if we look at, say, 660 millimeter light often used in PDM studies using that carrageen and paw edema model in mice.
The swelling model.
Right. They've shown that 660 millimeter laser treatment can improve the edema, reduce the swelling, and decrease the levels of inflammatory cytokines in the paw.
So tackling both the symptom and the underlying signals?
Seems so. And what's quite interesting, one study comparing wavelengths in that same model found that 660 millimeter light was actually more effective than another wavelength tested at reducing the hyperalgesia, the pain sensitivity.
Really? So 660 might have a particular knack for the pain aspect.
It suggests that, yeah, it hints that different wavelengths, even within the effective red NIR range, might have slightly different strengths or optimal targets, whether it's reducing swelling or directly impacting pain pathways.
Fascinating. It's clearly not just any light, it's specific light doing specific things. Now, what about the immune cells themselves, the ones actually doing the inflammation work, like neutrophils or macrophages? Does PBM interact with them?
Oh, absolutely. That's a crucial part of the mechanism. Let's take PMNs, polymorphonuclear cells, often the first responders, neutrophils being a key type.
Okay.
Research suggests that PBM, at certain doses anyway, can actually inhibit their proliferation, slow down their buildup at the site of inflammation, and also reduce the pro-inflammatory cytokines they release. This has been seen in arthritis models.
So calming down the initial shock troops, so to speak.
That's one way to think about it. Then you have macrophages. These are really interesting because they can kind of wear different hats.
Right, I've heard about M1 and M2 macrophages.
Exactly. M1 macrophages are typically pro-inflammatory. They drive inflammation. M2 macrophages are generally considered more anti-inflammatory and involved in resolving inflammation and tissue repair.
Okay, so you want fewer M1s and more M2s if you have chronic inflammation.
Ideally, yes. You want to shift that balance. And studies using PBM, for instance, with 780 millimeters or 660 millimeter lasers have indicated that it can do just that. It seems to help reduce the number of M1s and increase the M2 population.
So it's pushing the immune system towards a more resolving, healing state.
That appears to be a key effect. Modulating the macrophage phenotype is a really important anti-inflammatory mechanism of PBM.
That's incredibly sophisticated for just light. Okay, and the pain. We touched on hyperalgesia, but the pain that comes with the swelling, the edema, how does PBM help there?
Well, the pain reduction is often one of the most significant benefits reported, both in studies and anecdotally.
Mm.
PBM seems to tackle this on potentially a couple of fronts.
Okay.
Firstly, there's evidence that can directly affect nerve conduction.
Okay.
It might slow down the transmission of pain signals along nerve fibers.
Like putting a damper on the pain messages.
Kind of, yes. Secondly, by reducing those pro-inflammatory cytokines we talked about.
Right, the TNF alpha, IL-1 beta.
Exactly. Since those cytokines themselves contribute to pain sensitivity, reducing them naturally helps alleviate the pain.
So it hits the cause and the signal?
That's the idea. And going back to those mouse paw edema studies, they consistently show that PBM, using wavelengths like 660 millimeter or even 830 millimeter, significantly reduces hyperalgesia. The treated animals just tolerate more pressure or heat before showing a pain response.
And again, the specifics matter. Wavelength, dose.
Yes, different studies have found varying degrees of effectiveness for edema versus hyperalgesia, depending on the exact parameters used. It reinforces that optimizing the treatment is key.
Okay, so pulling all this together, it really sounds like the science is, well, pretty strongly indicating that photobiomodulation, especially using these specific wavelengths like 635 millimeter and 810 millimeter, is genuinely promising for reducing inflammation, easing the pain that comes with edema. And this isn't just theory, right? It's backed by a growing pile of solid research.
It really is. The evidence base is becoming quite substantial.
And given how many people struggle with inflammation and swelling, the potential impact here feels, well, huge. It's definitely exciting to see companies like MedWave Group exploring these PBM solutions.
Precisely. The compelling evidence we've walked through today really does highlight the potential. Now, obviously, research is always ongoing. Scientists are still working to fine-tune the optimal parameters, the best wavelengths, doses, timings for different conditions.
Sure, there's always more to learn.
Always. But the findings we already have offer, I think, considerable hope. Hope for developing effective and, importantly, often less invasive ways to manage inflammation, edema, and the pain they cause.
Yeah. You know, when you stop and think about how fundamentally our bodies interact with light, sunlight, circadian rhythms, it really makes you wonder, doesn't it? Beyond what we're looking at today, how else might we be able to harness this power? The power of specific light for, targeted healing, for well-being in the future.
It's a profound question. The potential applications could be vast.
Definitely something fascinating to keep an eye on. Well, thanks for joining us for this deep dive today.
My pleasure.